How's it going? It's Chow here again. Today we're going to discuss a little bit about proteins. Proteins are pretty interesting, I think, but this is going to be a basic bare bones version that's probably good for an intro biology course, maybe not so much for a biochemistry or organic chemistry class. But let's get started. So first of all, we have to discuss what exactly do proteins do. Well, they actually do a lot of things. So proteins can actually go through a lot of these mechanisms that can allow the body to go through certain processes. So some of these include enzymatic action. So you use an enzyme, for instance, to lower the activation energy. You can use proteins, for instance, for antibodies and recognition. So recognition of foreign materials, that's very important for the immune system. It's also very important for signaling as well as recepting. So you have receptors that are made of protein that can pick up certain signals and then it'll tell the cell what to do next. It could be used for transport, it could be used for structure, it can be used for activating catabolic pathways. For instance, if you think about epinephrine coming in and then binding to that G protein coupled receptor and then making that cascade for you to break down glycogen. So pathways and catabolic pathways, proteins can do that as well. So general regulations, both in hormone levels and genes, proteins can do those as well. So what exactly can proteins not do? And the answer is there are actually a few things. And one of the key thing is actually energy storage. So proteins can't do the whole energy storage thing. That's more restricted to fats and sugars. So for instance, you can store energy as triglycerides, as fat, as adipose tissue. And you can store also energy as glycogen, where you can break it down to glucose and use that. But proteins aren't really good when it comes to storage for energy-specific purposes. It also can't be used really to store information. So remember that when it comes to storing information, that's more of a DNA thing, or you can perhaps even talk about RNA in some organisms, but it's nucleic acids that store that genetic information, not proteins. But proteins are pretty interesting, and they're very crucial and critical to the body. So when we look at proteins, we obviously have to look at the individual structure of proteins themselves. So these little tiny small component structures of proteins are called amino acids. So generally speaking, there are 20-ish common amino acids, technically more if you look at some of the other species and lineages out there like archaea. But in most vertebrates, you see 20 main key amino acids. Now, the amino acids are actually quite similar in many ways. They all have an amino group over here. They also have a carboxyl group over here. And the one thing that really differs between individual amino acids is not the amino group or the carboxyl group, but this R group over here, sometimes called a radical group. And this is what's different between different amino acids. So you can have a glycine or a serine, and the backbone, which is the amino group, this alpha carbon over here, the central carbon or alpha carbon, and the carboxyl group, that's all the same, but it differs within the R group. So there you go. And then of course, amino acids also have optical isomers, and so depending on how the carboxyl and amino groups are structured, you can actually have variation within that structure. And this is something that you'll probably talk about in organic chemistry. So if you're taking orgo, you'll know what I'm describing. But it's basically looking at how everything is structured. What is the rotation, the optical rotation? And we have L conformation and D conformation. Biologically speaking, most proteins are in the L conformation. That's a little bit different in sugars. Because of sugars, it's in the D conformation. But in proteins, the L conformation is the biologically active conformation, whereas the D conformation is not. The other thing about proteins that's very important is that they can be stripped of certain hydrogen ions, for instance, and you can create different charges. So what a zwitterion then is that it has both a positive charge and a negative charge. Again, if you're taking organic chemistry, you might have discussed zwitterions. But it's the state where you have a protein strand. It doesn't have to be just an amino acid. It can actually be a protein, but in this case, we're using an amino acid for simplicity's sake. You can see that in this amino acid, we have a positive charge on the amino group and then a negative charge on the carboxyl group. And what this causes is a positive charge and a negative charge, and they cancel out to zero. So a Zwitter ion is one particular ion that has a positive charge and a negative charge, but it cancels out to zero. And this can have very strong implications for what exactly the protein is going to be like. Is it going to be soluble at a certain pH? And this is more of a biochemistry topic, but you can then talk about what's the isoelectric point, where is it most neutral, what pH is it necessary to keep that protein neutral, etc., etc., etc. But this is a good way potentially to look at specific amino acids, and then if you combine those amino acids into a big chain, you can still use this concept of the Zwitter ion to basically help you understand which pH you want to keep a certain protein at, or perhaps what pH is this protein most active at doing whatever it's doing. So there you go, amino acids. Now, if we're talking about amino acids, obviously we can't just discuss amino acids nonstop. We have to group them. And amino acids are grouped in a couple of ways. There are different methods of grouping, but one of the key ones that are often seen in a biological classroom is this particular grouping method. You have nonpolar side chains, so you put the ones with nonpolar side chains together. You can put the ones with polar side chains together, and then you put those that are electrically charged in a separate group, and you can split them up based on basically what pKa they're going to be that's going to make them negative and or positive. And that can cause acidic ones or basic ones. So obviously your nonpolar side chains are the ones that are generally hydrophobic. Your polar side chains are generally the ones that are hydrophilic. And then also acidic and basic ones can potentially also be fairly hydrophilic, but it may be dependent on the pH as well. So acidic ones include aspartate and glutamate, and you have your basic ones at neutral pH that include things like lysine and arginine and histidine. So there you go. So this is probably one of the initial ways to kind of break down your amino acid groups. And in terms of memorization, usually they ask you to memorize probably like 6 to 8 of them. And that's something that you probably should ask your professor or listen closely in lecture to see what you need to memorize. Oftentimes it's something like glycine or alanine or phenylalanine, which is over here, or it can be something like lysine, which is down here. Sometimes they ask you to memorize aspartate or maybe even something like serine, for instance. So just keep an eye out and see what you need to memorize. This is another way of grouping. So obviously similar structuring as before, except we're making a special case over here with the cysteine, the glycine, and the proline. Obviously the glycine is kind of unique in the sense that it only has a hydrogen as that R group. The proline is a little bit strange because it forms a bond, so the side chain branches out and actually forms a bond to the amino group. And then cysteine is a little bit unique because it can form disulfide bonds. So cysteines can bond together and the sulfurs can form a bridge. And this can have very strong implications on protein structure. So there you go. So when it comes to understanding amino acids, what exactly do you need to do? Well, first of all, you need to question yourself and look at that amino acid and see whether it is charged or uncharged. So what you do is you obviously look for positive or negative R groups, and obviously, you know, the positive thing likes to go with the negative thing, and so positively charged amino acids like to form some kind of an interaction with negatively charged amino acids, and of course vice versa. The next thing you can look at is whether it is polar or nonpolar. Oftentimes nonpolar amino acids have kind of long hydrocarbon chains, and polar individual amino acids often show some kind of a functional group that has electronegativity or elements that have been either completely reduced to be positive or completely stripped of its ions in a certain way to make it negative. Now, the other thing that you can look at is whether it's hydrophilic or hydrophobic. So again, hydrophilic forms some kind of a strong bond or interaction with water. Those are your charged or polar amino acids. And then your hydrophobic amino acids are the ones that don't really form too much of an interaction with water. And that's going to end up being kind of your non-polar amino acids. And of course, we can look at those special cases again, as I mentioned earlier, where cysteine can form those disulfide bridges, which can have very strong implication for structure. Glycine is pretty small, and the R group is just a hydrogen. And of course, proline forms that weird ring structure, which can potentially limit flexibility, and it can cause potentially like cis formations instead of a more perhaps favorable transformation. So it can have limits on its ultimate flexibility. So there you go. So let's talk a little bit about the structure of proteins. Now, proteins come in 4 main structures. You have primary, secondary, tertiary, and quaternary, right? So primary is probably the easiest one because it is just the chain of amino acids. There's no folding, no hydrophobic interactions, not really a lot of hydrogen bonding. It's just that chain of amino acids. Now, in order to form that chain of amino acids, you need to run through this thing called a condensation reaction. So it's sometimes called a dehydration synthesis reaction because you're synthesizing something via the removal of water. So you can see over here, right, you have 2 hydrogens and then an oxygen over here. And by removing, so by taking that oxygen and those hydrogens, you can form water. And water leaves, and then of course you have a peptide linkage that forms. And this is generally done in the ribosome with, I believe, peptidyl transferase. And it can be quite a fast-moving reaction, and it goes through really, really quickly, and you can form this long chain of amino acids. But that single linear chain of amino acids is what's designated as the primary structure. And it is going to have a repetition of various residues, I guess is what I should be saying, various residues, various amino acid residues. But what's really key here is that it's still a linear chain without any sort of folding or side chain interaction, none of that, just a single linear chain. So that's primary structure. As you move on, you can get into secondary structure where you're going to start seeing some of these side chain interactions. There are 2 types of secondary structures that are very commonly found in proteins. The first one is the alpha helice. So the alpha helix is just this coiled kind of corkscrew shape, and the alpha helix is often formed by interactions of different amino acid residues about 4 units apart. So for instance, this particular carboxyl group over here, this carboxyl group over here will likely interact and hydrogen bond with this amino group over here. So 1, 2, 3, 4 residues down. And that's what often happens in an alpha helix, and it forms a corkscrew-like coil. The other type of secondary structure is the beta-pleated sheet, and this is just interaction of the side chain of 2 different sheets of protein. I believe you can actually have an instance where it's both parallel and anti-parallel. And depending on whether it's anti-parallel or parallel, it can be favored or less favored. So in an anti-parallel sense, as shown over here, there's going to be more favorable pairing. Whereas if it's parallel, it can still pair into a beta-pleated sheet, but it's less favorable. That's more of a side trivial information, so it's not as important. But do keep in mind that in terms of secondary structure, we're seeing hydrogen bonds coming in, and it's often with interactions of those side chains, that R group. So moving on, we can go to tertiary structure. And tertiary structure is this part where we start seeing that protein folding into more of a three-dimensional shape. Instead of just a sheet or a coil, it's actually folding upon itself to form kind of a glob. So the bending and folding causes a macromolecule with a specific 3D shape, and obviously the protein has to be folded into a specific shape to allow for a specific function. Tertiary structures often express, so it's often between R groups, and there are often these types of common concepts that are seen within a tertiary structure methodology. And the first one is the disulfide bridge. So this is with two cysteine molecules, again, with those sulfurs forming those bonds together and linking various parts of that protein, and it can ultimately form this giant connected structure. Hydrogen bonds are still somewhat important in the tertiary structure, albeit perhaps it's maybe more important for secondary structure interactions. You have aggregation of hydrophobic side chains, so hydrophobic interactions are very important in a tertiary situation. And then, of course, you have van der Waals forces as well as ionic interactions between, for instance, over here, you have maybe a bit of salt bridge formation between one group that's positive and one group that's more negative. So many of these interactions are found in the tertiary structure, and the protein ultimately starts folding up. So after that, you can actually have multiple associations, so multiple subunits, as they call it, come together. So tertiary subunits that bind together into a quaternary structure. So a quaternary structure is often consistent of multiple chains, so multiple proteins, you can say, coming together, multiple polypeptide chains coming together and forming this giant group, and that's the quaternary structure. So again, subunits are generally individual peptides, often individual proteins, and perhaps in many instances they also have their own unique primary, secondary, and tertiary structures. And of course, with quaternary structure, much like tertiary structure, you can have these disulfide bond interactions. So one subunit can be connected to the other subunit through disulfide bonds or disulfide bridges. So there you go. And once again, that disulfide bridge that connects various subunits together, or maybe just causes that one subunit to fold in the tertiary structure, is caused by the amino acid cysteine. Hemoglobin. Hemoglobin is a very common tertiary structure individual subunit that comes together to form a quaternary subunit. So hemoglobin is actually a great representation of all four levels of protein structure. And it actually is a quaternary protein glob. So you have four independent protein units or protein subunits, and you actually need all four of them in order for proper oxygen binding, mediation, oxygen release, and in order for essential function, you have to have all four be together. So if you remove one of these subunits, it doesn't do as well. So there you go. Now, when it comes to proteins, obviously it doesn't have to stay exactly the way it is. You can break down some of those proteins, and this is in a process called denaturing. Now, a protein is generally considered denatured when the secondary and tertiary structure is broken down. The primary structure with those peptide bonds, which are covalent bonds, are very, very strong, and so they're going to be much more difficult to just simply denature and break apart. Basically, when it comes to denaturing, we're talking about the breakage of secondary and tertiary structure. When it comes to denaturing, there are lots of things that can cause a protein to denature, and some of the things include temperature, pH, maybe high concentrations of certain polar or maybe non-polar substances. And what's interesting about denatured proteins is that in many biological systems, you can actually return it to normal by bringing the various conditions back to normal. So for instance, if you raise the temperature really high and the protein gets denatured, if you lower the temperature slightly bit by bit back to what it's supposed to be, the protein can actually often refold on its own to relatively strong accuracy. Sometimes that requires a bit of help, so you need some extra proteins to come in to help it refold. But unfortunately, there are instances too where there are proteins that become denatured and therefore can't actually refold on their own and really just can't refold at all. And so some of these denaturings are indeed irreversible and permanent, and that can have lethal and potentially dangerous effects on whatever organism that protein is part of. But when it comes to proteins, obviously we talk about things like reactions and catabolism and anabolism. And some of these reactions involve proteins called enzymes. And with enzymes, they require often a very specific shape. So a specific enzyme has a specific shape, and it reacts with a substrate that kind of fits into that particular enzyme. And in the past, we call it the lock and key kind of analogy, where it's exact fit, but really today with more research, we understand that it's not so much a lock and key analogy, but more of a induced fit analogy. So instead of, lock and key, it's more like putting on a latex glove or something like that. It's not quite 100% matching, but you can induce a fit and then ultimately make your protein substrate complex, or more specifically, a protein enzyme complex. So what's really important from maybe a biochemistry perspective is that you need those R groups on the surface to be able to interact through ionic, hydrophobic, or maybe hydrogen bonds with whatever substrate you're working with. So there has to be a level of specificity involving enzymes. So that's basically it for most of the introductory information to proteins. Obviously, if you're taking something like biochemistry, you're going to go into a lot more detail than what's covered in BI 108. But one of the things that perhaps is important is to memorize those amino acids that your professor asks you to. The other thing might be to get a good understanding of what is the definition of a hydrophilic amino acid, what is a hydrophobic amino acid. So if they give you an amino acid that you're supposed to memorize, for instance, like maybe glycine, be able to recognize whether that amino acid may be put in the outside of the protein, or maybe is it going to be shuffled into the center when it's folded. So maybe keep an eye out for those as well. But ultimately, proteins are, again, very important and critical molecules within biological systems. And they can perform a lot of functions. And while sure, they can't store energy or store genetic information, there are a lot of things that proteins can do. And perhaps it's this ability for proteins to react to certain specific conditions and all these characteristics and traits that they can go through to allow for the body to function that makes proteins extremely interesting and perhaps worth pursuing. So I hope this basic introductory information is actually useful, and I hope all of you had an enjoyable time, and I will see you all next time. And as always, good luck studying, everyone.